Daniel Ayo, Moses Mwanga, Evelyn Nansubuga. So thank you very much, ladies and gentlemen. We are going to move into the session for malaria. My name is Dr. Adoke Yeka. Not Kabaka Yeka, it's Adoke Yeka. I'm a member of faculty at Makerere University School of Public Health. We are going to have a very interesting session on malaria. I think you have had previous sessions, but this is going to be more interesting. We are going to look at different aspects of malaria. There are about 8 interesting presentations lined up. We'll look at malaria species diversity. How is it in our setting? I think we have been used to the talk of always having Plasmodium falciparum. Are we having other species? What is the implication of having those species in our setting? And then we'll be having presentations about recurrent malaria, repeated infections of malaria. What are they? What is their significance? And we'll dive deeper, going to look at molecular aspects of malaria. The gene deletions, do they have implications on how we diagnose malaria? Do they have implications on the tools that we use for malaria diagnosis? We'll also move to look at drugs used for preventing malaria in any important group of people, pregnant mothers. Which is the best drug? What works? What doesn't work? So please come around to listen to these interesting discussions. And we'll look at immunity. The body's acquisition of immunity. What does hinder it? Certain things which may happen at the genetic level may hinder the development of immunity. And some of those have important implications on current control tools like the malaria vaccines. So be around to listen to this. We'll also look at malaria transmission, gametocytes. Are the key forms of parasites that are responsible for transmission. So a lot of talk about gametocytes, the infectivity and so on, and a bit of touch about drug resistance. So we have a panel before us who are going to make the presentation about this. And to each member of the panel, we are going to give you 8 minutes to make your presentation. So please keep time. And I'm going to remind you when we have 2 minutes left to make your presentation. Innocent there? Yes, our first presentation will be from Innocent. Innocent, the other name is a bit difficult to pronounce, but I hope I pronounce it properly. Innocent Wiringirimana. If I pronounce it badly, excuse me. So Innocent is a laboratory technologist and is a research scientist who has been working in Uganda for quite some time with the infectious diseases research collaboration. And a lot of his work is primarily oriented around malaria research, looking at genomics and looking at information on disease surveillance. He has done a lot of relevant research which links laboratory genomics and the public health outcomes. So ladies and gentlemen, let us welcome our first presenter, Innocent. Innocent, you're most welcome.
Pointing the other side. Okay. Okay. Yes, ladies and gentlemen, good morning. My name is Innocent Wiringirimana, and I work with Infectious Diseases Research Collaboration. Today, I'll be presenting prevalence of and factors associated with mixed species infection in patients diagnosed with falciparum malaria in Uganda. As we all may be aware, yes, as we all may be aware that Plasmodium falciparum continues to be responsible for most malaria-related morbidity and mortality in Uganda. However, its co-infection with other species such as Plasmodium malariae worsens disease and continues to increase PF gametocytemia, while its co-infection with Plasmodium ovale and Plasmodium vivax can cause a relapse of infection. These non-PF species continue to be in our population by mere fact that they're always missed by traditional diagnostic methods like rapid diagnostic kits and microscopy, partially because they occur usually in low parasitemia, but also most of the RDTs that we have on market in Uganda are detect only PF. But the good news is that with the emergence of molecular techniques, molecular diagnostic techniques, we are able to detect these mixed infections reliably and accurately. So understanding the prevalence and determinants of these mixed Plasmodium species infections becomes very vital for effective diagnosis, treatment, and malaria control efforts. Therefore, this study aimed at determining the prevalence of mixed Plasmodium species in patients diagnosed with malaria in Uganda, and also to identify factors associated with presence of mixed Plasmodium species infections in Uganda. A total of 200 dry blood spot samples were collected twice yearly in high transmission sites, and we also had an all-year round collection of samples from low transmission sites. And we did this in a way that we could select sites covering the entire country uniformly, as you can see on that map. The samples that were collected were transported to National Malaria Reference and Research Laboratory at CPHL, where DNA extraction was done, and samples that were positive for PF by various qPCR were sequenced using multiplex amplicon sequencing protocol called MAD4HATER. This was developed by one of our collaborators and validated here in Uganda by us. And we did identify non-falciparum species by, of course, identifying if there was amplification of any of the targets of PM, PV, Plasmodium norisii, and any of the two subspecies of Plasmodium ovale. A total of 5,213 samples were sequenced, and overall we had a prevalence of mixed Plasmodium species infection as 7.6%, which accounted for a total of 396 samples out of the 5,213 that were sequenced. To note, the most prevalent mixed infection was PFPM, accounting for 4.1% of the samples that were sequenced, followed by PFPO. There was a very low prevalence of PFPV, which only accounted for only 2 samples of the total samples that were sequenced. And when we go to the individual sites, we noted that the prevalence of mixed Plasmodium species infection ranged from 1.7% to 22.3%. But to note is that the median prevalence was at 7.3%, and our interquartile range was 4.4 to 11.1, which points out the fact that these mixed Plasmodium species infections are not occurring uniformly in different sites. We have sites where they are highly prevalent, and other sites they are relatively low or no mixed species infections. We also noted that there was an association between complexity of infection of PF with mixed species infection, where after adjusting for some of the possible co-founders such as parasitemia and age, we had an adjusted prevalence ratio of 2.26, particularly for those samples that were showing a COI of 2 to 5 of PF. And even when we tried to consider the group that had a higher COI of greater than 5, we saw that the APR was increasing further to 4.79, which shows a stronger relationship. We also noted a strong positive correlation between site-level microscopic parasite prevalence, which was assessed by a cross-sectional community survey, and this was also, there was a positive correlation between it and mixed species infection with a Spearman correlation coefficient of 0.69. Yeah, this was indeed stronger with PFPM than PFPO. So what are our conclusions? We conclude that mixed Plasmodium species infections show relatively higher prevalence in some regions of Uganda. And to note is that PFPM co-infection show highest prevalence of all these, followed by PFPO. And this is key or mostly seen in high transmission settings. Mixed species infections are positively associated with the transmission intensity and complexity of infection of Plasmodium falciparum. The limitation that we had in this study was that low transmission sites had fewer samples that were collected because of the lower burden of malaria in this area, and very few participants could meet the recruitment criteria. And of course, this ended up exaggerating the prevalence that we are noting in these sites in case a few samples showed mixed infections. The study implications are that, of course, we shall need a cross-sectional survey to determine the prevalence of both mixed and pure non-PF infections if we are to establish the overall burden of non-Plasmodium falciparum species in Uganda, since this study only focused on mixed infections. We also see that since the COI is associated with mixed infections, then its monitoring will be very vital for us to predict the possible areas at risk of mixed infections. And after that, it is also important to have targeted control measures in high mixed infection regions if we are to reduce both PF and mixed species infections. And so, like we had earlier seen, also enhanced species-specific treatment guidelines will be very important since we know that these infections can complicate drug efficacy, also put the patient at risk of infection severity and relapses. And in conclusion, I will acknowledge infectious disease research collaboration, Bill and Melinda Gates Foundation for sponsoring the IMAS-U study under which this project was done, the University of California, San Francisco, our collaborators, and the National Malaria Reference and Research Laboratory at the Central Public Health Laboratories of the Republic of Uganda, the lab where this work was done from. Thank you so much for listening.
Thank you very much, Innocent, for that presentation. I think he's highlighted important issues about malaria co-infection. Now we know we do have a lot of mixed infections, up to 7.6%, and they do have public health implications. Having mixed infections do have public health implications, as he pointed out, like increasing transmission intensity and complexity of infection. Can you give another round of clap for the presenter? Now we are moving to our second presentation. Okay, we now invite Ms. Ngimana to come and make his presentation. Most people you see here are from IDRC, and I'm proud to be associated with IDRC. And Sengimana is a Ugandan molecular lab technologist. He works with IDRC. He mainly specializes in the area of malaria genomics and also looking at diagnostics and the malaria drug resistance surveillance. His work bridges mainly advanced molecular laboratory techniques together with the public health relevant research. This supports the efforts in understanding and monitoring malaria parasite evaluation in Uganda. So ladies and gentlemen, let us welcome Sengimana to make the presentation.
Thank you, Dr. Eka, for the good introduction. Ladies and gentlemen, good morning. My name is Bienvenu Sengiman, and I work with Infectious Disease Research Collaboration. And today I just want to give a talk on the findings of our study that is entitled Screening for Plasmodium falciparum, HRP2 and HRP3 gene deletions in Uganda using a highly multiplex deep sequencing assay called MAD4ATA. To just give a brief background and rationale of our study, the deletions in the histidine-rich protein 2 and 3 cause false negative HRP2-based RDT results in the Plasmodium falciparum infected individuals, which usually leads to misdiagnosis and also improper treatment. And as we all know, is that these rapid diagnostic tests remain the Uganda's primary malaria diagnostic tool because of their ease of use and also affordability. But we have usually a problem where the antigen that we usually test in these RDTs, sometimes the parasites are not able to express them because of deletions in the HRP2 or HRP3 genes, which usually express those histidine-rich protein 2 and 3, which is usually the biomarker that we test in the RDTs. And these deletions have actually emerged with high prevalences in the Horn of Africa and now spreading with high to other parts across the continent, just like you can see on the map on our far right. WHO recommends switching to non-HRP2/3 based RDTs once these deletions actually exceed 5%. And our study was set to estimate yearly prevalence and characterize the geographical spread of these deletions in Uganda. For methods, study design and study preparation, so this study was nested in a bigger cross-section study called IMAS, and IMAS is implementing malaria molecular surveillance in Uganda, and it was carried out in the year 2023 and year 2024. So results are for these years, but the study is still ongoing. We collected approximately 200 dry blood spots samples from patients who were diagnosed with uncomplicated malaria from each sentinel site, so you can call them malaria research centers, which are spread across Uganda, just like you can see on our map on the far right. We have sites in different sub-regions of Uganda, covering almost uniformly the whole of Uganda, a total of 32 sites. There were 2 rounds of sample collections per year, and in the high transmission areas, just like it is indicated there, round 1 up to round 4. And for the case of road transmission sites, samples were collected year-round because we usually get few samples in such sites. So we just do a year-round sample collection. So for the laboratory procedures we carried out, after getting the dry blood spots in our laboratory, we did DNA extraction from them using twin Cherex 100 method. And after getting the DNA, we had to do its quantification and also confirming that we have Plasmodium falciparum, and we used the VAR ATS2 PCR. And then we selected high quality DNA samples, 100 for each site per round of collection. And we did library preparation for these samples, where we prepared our libraries using MAD4HAT assay, which has targets in diagnostic resistance, and these are the ones of now for our interest. Then it has also other targets in drug resistance, diversity, and also immune-related kind of targets. So we did the sequencing of the libraries, and the sequencing was done using next generation Illumina platforms, specifically the MySeq and NextSeq, which we have on our laboratory. For data analysis and interpretation, the sequence data was analyzed using MAD4HAT pipeline, which was used to generate array code data across diversity, drug, and diagnostic resistance loci. And for interest now is the diagnostic resistant loci. So the HRP3 deletions fall under mutation, which are called copy number variation. So for copy number variation analysis was estimated using generalized additive model, which usually corrects for amplification biases, corrects for biases in coverage, and we calculated for changes in read depths when we compare it relative to the control code PF3D7, which is known to have no deletions in these genes. So we CNV4 change threshold of 5 and below was used to infer samples as suspected deleted in the HRP2 or HRP3 target loci. And the prevalence and geographical distribution of these deletions was performed using our software. Diving into our results, we see that we had few suspected deletions in the HRP3 gene, and we did not observe any deletions in HRP2 gene deletions, in the HRP2 gene. And this was in the total of sample that was 7,524, and only 61 samples exhibited these deletions, which represents 0.8%. So just those pie charts below tries to give us a picture that is in the year 2023 and the year 2024, and we see the picture is almost the same, with this low prevalence of HRP3 deletions of just approximately 0.8%. But still, we wanted to also see, even if we have these low prevalences, whether we have differences when we do granular details, maybe at the sub-region level, and we see that HRP3 deletions prevalence varied by site in regions per year, with all the sites having less than 5% of these deletions. But what note is, for Kigezi sub-region, had sites with a greater number of samples with suspected the HRP3 deletions compared to other sites in other sub-regions of Uganda. So our study is in 10 out of 15 sub-regions, just as you can see in the map there. In conclusion and the study implications, we have a good news maybe for Ugandan setting, that these HRP2 and HRP3 deletions remain very rare in Uganda, hence a very limited threat to the usefulness of HRP2-based RDTs currently. And we want to note that Kigezi sub-region, which is a low malaria transmission area, had highest proportion of samples with suspected HRP3 deletions. And the possible explanation for this is because when the transmission tends to go low, we usually have low complexity of infection. In other words, we usually have either just very few parasites few types of parasites within a single infection. So we can easily pick out such kind of deletions. So for that reason, we can easily have elevated deletions when you are screening for them. So for the implications of our findings, we see that the findings of this study highlight that the HRP2 and 3 deletions do not pose a threat to the usefulness of HRP2-based RDTs in Uganda, which is good news. And cases of false negatives, like I mentioned, these deletions usually cause false negatives. We always need to investigate them, and want to highlight that when we are finding the reasons, usually they will not be due to deletions. So we need to find other reasons why we are having false negatives in case they appear. For the limitations, we see our results were interpreted with strict cutoffs, which could have caused to miss some deletions, especially in mixed or polyclonal infections, especially if the HRP2 and 3 gene deletions were in minor array in the sample, was a minor array in the sample. For recommendation, we recommend that continuous surveillance for these HRP3 gene deletions to continue in our setting, given that we saw some presence of few HRP3 deletions, especially in the southwestern part of Uganda. I'd like to acknowledge the team of Imazu indicated there in the picture and all the collaborating institutions, as you can see there in their logos, the developers and implementers of MADI4 at ASE, the study participants, and also the audience. Thank you so much for listening.
Yes, thank you very much, Nsengimana, for enlightening us about these gene deletions. There are some aspects of the genes. If they are not there, it makes our diagnostic devices not work. The RDTs we use for testing malaria. So at least we are relieved. The prevalence is just around 1%. But some other studies have shown higher prevalence, 2 to 3%, but we are still below 5% prevalence. So we can still trust results we get from our RDTs. So not to have false negatives when you actually have the disease. Thank you for giving us that assurance. But it's important that we continue with the surveillance. So ladies and gentlemen, let's once more give a big round of clap for Nsengimana for that presentation. Our next presentation is from an eminent scientist, Dr. Abel Kakuru. Dr. Abel Kakuru is one of the leaders, the directors for IDRC sites. The site in Tororo is the one leading that research center. And he has done a number of studies on HIV, number of studies on a prevention of malaria among pregnant mothers using either sulfadoxine, pyrimethamine, or diatomycin piperaquine. And is coming to enlighten us more to see, are the drugs which we are using to prevent malaria, are they working? Because there's concern that Fancida, there's a lot of resistance to Fancida, it may not work. Does it still prevent malaria among pregnant mothers? So Dr. Abel Kakuru, you are welcome to give us the insights.
Thank you so much, Dr. Eka, for that wonderful introduction. Good morning, ladies and gentlemen. So I'm going to present results of a double-blind randomized control trial that we conducted in Busia, Uganda. So in this study, we used diatomycin piperaquine plus sulfadoxine pyrimethamine for the treatment of malaria in pregnancy, and we compared it with either drug alone. It was a double-blinded randomized control trial. So a little bit of the background. The World Health Organization recommends intermittent preventive treatment of malaria in pregnancy with sulfadoxine pyrimethamine. Intermittent preventive treatment is you give a curative dose even when the person is not sick, because you want to prevent the infection or the disease. And this should be started early in the second trimester, not in the first trimester, because SP is known to be neurotoxic, so you don't want to give it in the first trimester. However, the efficacy of sulfadoxine pyrimethamine, that is SP, is limited by widespread antifolate resistance, which is very common in sub-Saharan Africa. That map on the right is showing the prevalence of antifolate resistance markers in sub-Saharan Africa. The first one is showing DHPS 540E, which is a proxy marker of quintuple. There are 5 genes that were first studied, and if you have those 5 genes, SP cannot be used for treatment. So the prevalence, the first map shows the prevalence of 540E, which is a proxy marker of those 5 genes. And you can see that Uganda falls in the red zone, which is 70 to near circulation percent, that is 100%. Then the second map on the right shows the prevalence of a 581G. That mutation, once it's there, you have total resistance of both sulfadoxine and then permethrin. They are always 2 drugs. So you can see that it's creeping up. It started in the southwestern part of Uganda, but now we are even beginning to see it in Tororo. Sorry, no, it's not moving. So because of this resistance, we started looking for alternative drugs to replace SP for IPTP. And the dihydroartemisinin piperacin, it's a highly efficacious ACT and has a long post-treatment prophylactic effect because of the piperacin component of the drug. It's a very good, promising alternative. And in recent trials, we conducted the meta-analysis of recent trials conducted especially in East Africa. Toro and Busia contributed about 2, and then some conducted in Kenya by our collaborators from KEMRI. We showed that DP was associated with better malaria outcomes. It clears parasites, it prevents symptomatic infection. However, despite the wonderful malaria benefits, DP does not improve adverse birth outcomes. It doesn't improve birth outcomes. And the main reason why the WHO recommends IPTP is to prevent birth outcomes, especially to improve birth weight. And we also saw that despite SP being a poor anti-malarial, it still improves birth outcomes. The children that were born to these mothers who are on sulfadoxine permethamine were more likely to, less likely to have small for gestation age, and then they were also 50 grams heavier, whatever that means for public health implication. So these data suggest that SP may have additional non-malaria benefits compared to DP, which is a very good antimalarial, but doesn't improve birth outcomes. So we went ahead. Sorry. So we went ahead and tested the hypothesis that combining these 2 drugs, one which is a very good antimalarial and one which has non-malaria benefits, despite being a poor antimalarial, we could improve birth outcomes, because there are adverse birth outcomes related to malaria, and then there are those related to non-malaria causes. So we thought combining both drugs, DP plus SP, could be better than either drug alone. So to test this hypothesis, we conducted a double-blinded randomized control trial of IPTP with dihydroartemisinin piperacin plus sulfadoxine combination versus either drug alone in Ugandan pregnant women. The primary objective of the study was to compare the risk of adverse birth outcomes between the 3 IPTP arms, and the second objective was to compare malaria outcomes between the IPTP arms. So the study was conducted in Busia district, Eastern Uganda, an area of high malaria transmission intensity. Pregnant women without HIV were enrolled at 12 to 20 weeks of gestation as determined by ultrasound, and were randomized to monthly IPTP with SP, DP, or DP plus SP. We initiated study drugs at 16 or 20 weeks, depending on the gestation age that the pregnant woman was enrolled. Then we saw these women every month, that's about 28 days. And during these routine visits, we would administer them drugs, and then we collected blood for detection of malaria by microscopy and qPCR, and then we did a CBC at 20, 30, 28 and 36 weeks of gestation to monitor for adverse events. We encourage these women to deliver in the hospital so that we are able to measure birth outcomes. Our primary outcome was a composite adverse birth outcome, defined as either having a spontaneous abortion, a stillbirth, preterm delivery, small for gestational age, low birth weight, or neonatal death. Low birth weight is when you're born with less than 2.5 kilograms or 2,500 grams. Secondary outcomes included malaria outcomes, which is the incidence of symptomatic malaria, prevalence of parastemia during routine visits, and placental malaria at delivery. So the results. So we screened 3,063 pregnant women, and 306 were excluded. We enrolled 2,757 women who are randomized, and they were randomized to equal numbers to the three treatment arms. Many of them were followed to delivery. Our follow-up would end just 4 weeks after delivery because we wanted to see also the neonatal outcomes. So those are the baseline characteristics. You can see that they were well balanced. The mean age in years was about 24 years. They are relatively young women, especially in upcountry settings. Majority were enrolled at 12 to 16 weeks, which is very early in pregnancy because that doesn't normally happen in routine care settings where women typically wait to hear the baby kicking and then they come to the clinics to attend antenatal. About a quarter of them were prim gravid. We would have loved to have many, but they were just about a quarter. And at baseline, about 37% of them had malaria detected by microscopy. And then when we used qPCR, it increased to about 70%. The prevalence of the mark of resistance, which I talked about, was about 99%, which was nearly reaching saturation. So these are malaria-related outcomes, DP versus SP. DP was very good at preventing malaria outcomes. It nearly eliminated symptomatic malaria, reduced courage of microscopic parastemia, and then improved anemia, and then also improved the risk of placental malaria compared to SP. So these are selected malaria-related outcomes stratified by gravidity. Here we want to show you that actually the prime gravid women are the ones who are getting more malaria compared to mild gravid, as expected. So the burden of malaria is much higher in prime gravid compared to mild gravid, and the relative reductions of DP compared to SP were much greater in the prim gravid than mild gravid. So that is the risk of a composite adverse birth outcomes comparing SP with DP. You can see that actually SP was doing better than DP, and it was statistically significant. DP was associated with a 17% increase in the risk of a composite adverse birth outcome. So that is adverse birth outcomes, individual adverse birth outcomes stratified by gravidity. You can see that preterm birth was associated with about 60% reduction in the incidence of preterm birth, the risk of preterm birth. It's very, very important. And then when we looked at the other composite adverse birth outcome, the DP was associated with an increased risk of a composite adverse birth outcome, and this was mainly driven in the mild gravid than prime gravid. So that is comparing DP DP plus SP versus SP or versus either drug alone, you can see that DP plus SP was associated with an increased risk of a composite adverse birth outcome, but the difference was not statistically significant. We expected some reduction, but there was no reduction. So in conclusion, our study shows that the burden of malaria was remarkably high in women living in this high malaria transmission setting, given IPTP-SP, and monthly IPTP-DP was associated with a reduced burden of malaria, and these impacts were greater in prime gravid, and IPTP-DP with SP versus DP on adverse outcomes also differed by gravidity. DP was associated with the risk of a lower preterm birth, although this was not only in prime gravid, it was high overall, because if you look at interaction term, it was not statistically significant. But in multigravid, DP plus SP was associated with a high risk of small for gestation age and low birth weight. So combining DP with SP did not improve malaria outcomes, and we think this could be because maybe DP has an adverse effect on fetal growth. And then also we observed drug-drug interactions between these two drugs. We didn't expect them to interact, but we found an interaction, and we think this interaction could have contributed to these findings that we see. So caution should be taken when generalizing our results, because they were conducted in a very high transmission setting. So we need to conduct it in moderate transmission and lower transmission settings. So what are the study implications? We need to consider DP as an alternative for preventing malaria in prime gravid, because it shows that it's actually beneficial. And then DP plus SP should not be combined for IPTP, as this combination did not improve malaria or birth outcomes. We would like to acknowledge the following. Thank you so much for being a wonderful audience.
Yeah, thank you very much, Dr. Abel Kakuru, for highlighting these important issues. Of course, we have a big challenge. The current drug we use, Fansida, or sulfadoxine pyrimethamine, for preventing malaria in pregnancy, and what we use to improve birth outcomes, there's marked resistance to it. But the difference is in pregnancy, we are using it for prevention, not in people who are actively sick. But if you use it in people who are already sick, it doesn't work. But his results show that SP used alone still works in improving birth outcomes, which is good news. DP, if used alone, works very well among prime gravida. But if it comes to public health implication, can you use it alone just for a specific group? Those are things to think about. But one important caution, we do not have to combine dihydroartemisinin and piperacin with the sulfadoxine with the SP, because the combination doesn't work very well. Thank you, Dr. Abel, for presenting these important results from this study. Another round of clap for him. Yeah, now we are moving to our next presentation. The next presentation is from Daniel Ayo. Daniel Ayo? Yes, Daniel Ayo is also a Ugandan-based malaria researcher, and he is a study coordinator with infectious diseases research collaboration. As I said earlier, most of these people are from IDRC. He mainly specializes in malaria genomics and also studying malaria diagnostics and also malaria drug resistance surveillance. So Daniel Ayo is going to talk to us about cells which are important in immune functions in the body. So let us keenly listen to his presentation. Daniel, you are most welcome.
Thank you very much, Dr. Yeka. Good morning, ladies and gentlemen. My name is Daniel Ayo. Today I'm going to share with you some results from a study that we are doing in Kalongo about gametocyte production and infectivity among Ugandan uncomplicated malaria patients infected with Plasmodium falciparum parasites with reduced sensitivity to artemisinin. Yeah, just to give you a background, Plasmodium falciparum with reduced sensitivity to artemisinin recently emerged in Africa. This is associated with mutations in CLEH13 gene. And in Uganda, the common mutations that has been discovered or identified, C469Y and A675V, in addition to C469F. And then there has been a discussion to block the transmission, a case for use of primaquine to reduce the spread of mutant infections. So this study is carried out in Kalongo, in northern Uganda, where the malaria transmission is very intense. And then this is a site in Uganda where these mutations were first identified. So as you can look at the map of Uganda, those dots are the points where the mutations were identified. And looking at the World Health Organization scale, C469Y and A675V being the validated markers of artemisinin resistance. So I want to just let you know that in malaria transmission, mosquitoes pick gametocytes from humans and then inject sporozoites to humans. So our objectives were to determine the prevalence of CLEH13 mutations and then look at the gametocyte carriage, production, and all this by molecular methods. So we also wanted to look at the transmission potential of the PFK13 mutant infections, and then its association of these mutant infections with antimalarial use. So this is the summary of our selection criteria. We do a single time point study in uncomplicated malaria patients, 2 years older, no danger signs. And then we also deal with EDTA blood, where we quantify gametocytes and do separation of ring stage parasites. And then we also get the heparin blood for mosquito feeding assays. And also we look at lumefantrine measurements in randomly selected wild type and mutant infections. So this is our site. We do mosquito feeding assays with locally reared Anopheles gambiae, SS, molecularly confirmed. And then we do mosquito feeding using whole blood. And then we concentrate gametocytes 6 times as compared to whole blood. So that is a setup of our insectary. Yeah, just to let you know that we rear these mosquitoes from larvae, and then we do dissections of the midgut to look for the oocyst, which is a pre-infective stage of the malaria parasites. And then you see the setup of the transmission using the membrane feeding system. So from the samples that we collected, we want to show you that the PFK13 mutations is highly prevalent at low densities. And so in this case, we enrolled 235 patients with uncomplicated malaria. Then we did the sequencing of the propeller domain of the CLEH13 gene. And so from this, our study shows that 66% of our participants had wild-type parasites by sequencing, and then only 6% were pure mutant. So in this case, the C469Y or A675V. So 28% by sequencing were wild-type parasites, and then the mutant. And also to note that after sequencing, we found that 83% of this population had multiclonal infections, as you can see from the graph on my right. So we wanted to improve the sensitivity to detect this mutation. We developed the digital droplet PCR, which is very sensitive, using the in vitro samples, and then we applied it to this population. So from this, we found that 34% of the samples which were wild type, the 34% only were wild type, as compared to 66% by sequencing. And also we note that 6% remained pure mutant, and then the mixed population were 60%, as you can see from the graph on my right. So on the y-axis, I have the proportion of the mutant parasites by droplet PCR, and then we focus only on C469Y and A675V. So on the x-axis, I have the mutation group. I have the pure wild type, which is at 66%, and then the mixed population of the mutation in the middle, and then the pure mutant on the right. So the colors are coded by the genotypes. Yeah. So when we did gametocyte production and carriage, we found that the gametocyte production is slightly higher in the pure mutant as compared to the wild type, and we grouped them in different percentages. So we have the percentage of the mutant parasites on the x-axis, and then the gametocyte densities on the y-axis. So the 10%, this is total parasite biomass in the sample, and then you see that the 50% and above mutant parasites, they have a higher density of gametocytes. And so this was done by looking at mature gametocytes, considering the male and the female gametocyte transcripts, and all this by digital droplet PCR. So I want to take you to the most informative part of this study, where we looked at gametocyte carriage and mosquito infectivity. So we conducted a total of 454 mosquito feeding experiments using the membrane feeding system. So we dissected the mosquitoes 7 to 9 days post-membrane feeding for oocyst development in the midgut. From this, using the whole blood samples, 9.7% of the experiments with whole blood resulted into at least one mosquito getting infected, and then 1.4% in the magnetic sortings. So when we concentrated the gametocyte densities by increasing twice the concentration, the infectivity increased to 15.4% in the experiments, and then that is 2.4% of total mosquitoes becoming infected. So when we model this, the mosquito infectivity also correlates with the increase in the gametocyte densities. So after adjusting for gametocyte density, mosquito infection rates were not different between the wild type and then the mutant infections. So when we looked at the prevalence and the prior use of ACTs before clinical diagnosis, 37.8% of patients had detectable lumefantrine concentrations in plasma at enrollment, and there was no significant difference between the wild type parasites and the mutant infections in lumefantrine positive populations. So to conclude, the K13 mutations are common and mostly C469Y and A675V. Then the PFK13 mutant parasites are as transmissible as the wild type parasites. And so the study also showed that there's some signals of established artemisinin partial resistance circulating locally, since it can be transmitted by mosquitoes. So also multiclonal infections are common. The study implications here, there is high prevalence of multiclonal infections, which suggests intense transmission in the community due to highly diverse parasite clones. And then there's self-medication. Self-medication is common, and partial dosing of artemisinin combination therapies in the region. This may increase the likelihood of parasites becoming less susceptible to the current ACTs. Since the parasites with partial resistance to artemisins are common, we must test, treat quickly with effective medicines, and then also to strengthen the routine molecular surveillance and continuous K13 genotyping with rapid feedback to the districts. With all this, I want to thank our collaborators, Dr. Ambrosoli Memorial Hospital, where our insectary is established, Radboud University Medical Center, where the main funding comes from, my mother organization, IDRC, UCSF, and then, I thank the funder, the Dutch Research Council. Thank you.
Yes, thank you very much, Daniel Ayo, for giving us this important information. We know there's the problem of emerging resistance to artemisinin-based combination drugs, which we currently use to treat malaria. And the findings show that parasites which have the K13 mutations are the ones which are likely to exhibit resistance to antimalarial drugs. And this study clearly shows us the prevalence is high in the area where they did the study in northern Uganda. Another worrying thing is that parasites which are mutant or resistant, they are also highly transmissible. So you are likely to transmit more resistant parasites. So giving us an important message that malaria should be properly treated, such that we don't give a chance for emergence of resistance, because those parasites are highly transmissible and the resistance comes with a lot of implications. Thank you very much, Daniel, for highlighting this. A round of clap for Daniel. So we are moving to our second last presentation. Ladies have come last this time. I don't know how the organizers made this. Yeah, so we are going to welcome Evelyn Nansubuga. Evelyn Nansubuga is a biomedical laboratory technologist who is also working with infectious diseases research collaboration. She mainly specializes in immunology and also on molecular work related to malaria. So she's going to tell us about some cells involved in the immune process, the natural killer cell phenotypes and their functions in children. Evelyn, you are most welcome.
Good morning, everyone. I'm Evelyn Nansubuga, and I'm affiliated with IDRC. As my past presenters have presented in genomics, molecular work, bioinformatics surveillance, I'll be talking about an interesting topic, mainly in immunology. This is the host immune response. So we do know a lot of cells involved in promoting immunity. We know T cells, B cells, many innate cells, and I'll be mainly describing and talking in detail about natural killer cells. Natural killer cells, these are innate lymphoid cells, and they have been mainly studied in viral infections such as HIV, cancers, and I'm mainly talking about them in malaria. So my presentation is on the characterization of natural killer cell phenotypes and function in children with symptomatic and asymptomatic Plasmodium falciparum infection in a high transmission setting. So the background of my study as to why I chose these NK cells, a study done by our group in IDRC at Tolab discovered a certain subset of cells. These are CD56 negative NK cells. NK cells present in various forms. They have CD56 and 16 expression. So there is a variety of cells that do not have CD56 expression, and they are the adaptive NK cells. These NK cells have been, these CD56 NK cells have been studied to have high parasite clearance and decreased odds of symptomatic infection. And in this study, some areas were unexplored, such as studying longitudinally, understanding, studying the same child over a long period of time, pairing the infections, symptomatic and asymptomatic, in the same child, and also studying the cytokine stimulation effects upon leading to cytokine production. So the study objective is first to understand the NK cell phenotypes, the counts in the same child, and also to understand the cytokine, the production of cytokines upon cytokine stimulation. So the methods for my study, for this study, we selected samples in the musical cohort. Musical, it was a surveillance study that was done in 2021, leading to 2023, where children were selected to study uncomplicated malaria over a long period of time. For my study, I selected children in the 3 to 10 years old presenting with both symptomatic and asymptomatic parasitemia. Samples that are available for baseline, these were 14 days before the infection. Then day 0 is the onset of infection when the child presented with the infection. And day 14, 14 days after the infection. For this, PBMC samples were collected. PBMC stands for peripheral blood mononuclear cells, and onto which assays were performed, such as cytokine assay and phenotype assay. So the results for the study, we saw that the NK cell subsets, especially the CD56 negative, that are known to be very adaptive and protective in children, were actually mainly abundant in the asymptomatic malaria compared to the symptomatic malaria. As I mentioned before, asymptomatic malaria means absence of symptoms such as fever, and symptomatic malaria, presence of these symptoms such as fever. So these protective phenotypes and NK cell subsets were highly abundant during asymptomatic malaria. This strikes off our first objective. And then understanding still in depth in our first objective, understanding the NK cell counts and phenotypes. We studied also the adaptive markers present on these NK cell counts and subsets. So on analysis, this CD56 negative had highly expressed CD85J, and also the CD56 highly expressed NKG2C. These CD85J and NKG2C are some of the adaptive markers present on NK cells. So these illustrate such a protective function on NK cells. And then something about KIA markers. KIA markers are also, these are markers on NK cells, mainly adaptive NK cells. KIA stands for killer cell immunoglobulin receptor markers. KIAs can present in forms such as inhibitory and also activatory. And there's also a rare subset that has heterogeneous, both inhibitory and activatory. So those with inhibitory present with the L and activatory present with the S. So while analyzing these cells, we realized that these adaptive NK cells actually had the heterogeneous markers which present with both the inhibitory and activatory. This is the KRTL2, L3, and S2. As you can see, they are both inhibitory and activatory markers. So these were present on the adaptive NK cells, mainly the DIM and then the NEG. As we can see, the brights, there is low expression of these KIA markers. This is because they are known as the naive NK cells, but shall see their function in the next slides. So I'm going to the next objective, studying the cytokine production upon cytokine stimulation. In NK cells, as I have mentioned before, there are three subsets. So in the first objective, I was mainly talking about the DIM and then the NEGs. This is because these are the protective and the adaptive. But then the brights are known as the naive. But they are naive, but then they also produce cytokines. These cytokines that we studied are interferon gamma, TN alpha, and IL-10. So as first we're considering time points, so baseline 14 days before infection, we see that there's high cytokine production during asymptomatic malaria compared to symptomatic malaria. Then at onset of infection, there's a sudden shoot of cytokine production in symptomatic malaria. And this we are shown bringing about the fever. And then 14 days after infection, there's a sudden drop, which is relative to the baseline. So this covers our second objective, understanding the cytokine production during symptomatic and asymptomatic malaria. So this brings about understanding in depth the phenotypes of these cytokine producing cells, which are the CD56 bright, and which we see there is low expression of these phenotype markers. And then for our conclusion, we see that there's repeated, there's differentiation of these NK cells in places where there's high transmission intensity of malaria, and therefore bringing about the protective nature against symptomatic malaria. So for our study implications, there is need for larger longitudinal studies to actually confirm the protective association of these CD56 negative NK cells, and then also to incorporate these NK cell responses in future vaccine studies. Vaccines are coming about, and we need to make sure they are efficient to work in these children. So I would like to thank the team from Stanford Medicine, Malaria University, and IDRC for their work in putting this work together. Thank you.
Yes, thank you, Evelyn, for that presentation. I think this has challenged us, isn't it? We are talking about different types of natural killer cells. Some are called this, some are negative, some are what. But you don't worry about that. So it is showing the sort of capacity that the country has. People who can study immunology, look at different types of cells, and so to understand better the response of the body to malaria infection and how these cells change as malaria progresses from asymptomatic, when you have no symptoms, to symptomatic when you have symptoms. What happens to the cells? How do they mediate the process? Understanding this will help us to be able to develop control interventions based on those. So basically, the message is that these natural killer cells, they are present. And as they are called killer cells, their work is to kill. They kill the parasites. And they change. They are high when somebody doesn't have the disease. When you are symptomatic, the parasites are there. They come in large numbers. But as the disease goes away, the numbers also go down. So the body responds to the infection. Thank you very much, Evelyn. And I encourage many more people to take that direction and become immunologists. Can you give her a round of clap? So last but not the least, we are going to have a presentation from Monica Mbabazi. Monica Mbabazi currently is a PhD student at Malaria University, and her PhD is in bioinformatics and genomics. She is also a research fellow at Infectious Diseases Research Collaboration. Monica's research interest is mainly in applying bioinformatics and genomic tools such that we have a better understanding of malaria transmission dynamics in Uganda. And she's going to tell us about the genetic diversity, transmission networks, and drug resistance in southwestern Uganda. Monica, you are most welcome.
Good afternoon, everyone. Monica Mbabazi is my name. And today I noticed everyone presented about high malaria transmission or malaria problems in high transmission areas. And today, for my case, I'm going to present something from a low transmission site. And the question would be, why do we even care to understand malaria in low transmission sites? So I'm presenting a work that basically is on genetic diversity, transmission networks, and drug resistance in southwestern Uganda, which is a setting of low malaria transmission intensity. Yeah, so malaria remains a major public health challenge in southwestern Uganda or in the entire world. But if you look at southwestern Uganda, it has a much lower malaria transmission than the rest of the country. And so the National Malaria Elimination Division is interested in region elimination. So understanding factors sustaining malaria transmission in these low transmission sites is crucial for us to better implement our interventions for elimination and control. So in this study, we basically set out to comprehensively characterize malaria cases and then understand the contribution of importation in these low transmission sites. And basically we used travel survey data and the genomic data to do that. So we focused on 3 sites, that is Nyuko, Maziwa, and Kamwezi, and these are sites that border Rwanda and DRC. Nyuko and Maziwa are very low transmission sites, and in these sites we attempted to collect all the samples that were confirmed to have malaria by microscopy between the period of January 2023 to June 2024. And we managed to collect around 234 samples. Then in Kamwezi, which is more of a moderate transmission site, we attempted to basically collect all the samples that were confirmed to be microscopy positive between a period of July 2023 to August 2023. And we managed to have 100 samples that were collected over this time. So basically for epidemiological data, we collected this information based on the 2 sources. That is the Sentinel site surveillance database, which we usually refer to as UMSP database. It is a National Malaria Surveillance Program database. And then we also administered a travel survey data that captured information on the travel status and where these individuals were traveling to and the reasons of travel. And so for the genomic data, these samples, the DBS were collected from these individuals that were confirmed to have microscopy positive, and then they were transported at CPHL for processing and sequencing, where we used the Mad Hatter that most of my colleagues have talked about, which is sort of like a protocol for genotyping and sequencing malaria. And so we did the bioinformatics analysis, but basically we focused on different markers, the diversity markers and drug resistance markers. And then we did the bioinformatics analysis, and then we also analyzed this data using the Mad Hatter protocol that has been developed by our collaborators at UCSF and then optimized here in Uganda. And so for the genomics, we looked at different matrices. We looked at the diversity matrices, and one of them was the complexity of infection, what we refer to as the COI. And in this, you're basically looking at different distinct parasites that an individual carries. And then we looked at the effective COI, which is basically an adjusted measure for the complexity of infection. And so we looked at relatedness, wanted to see how related these parasites are. And we used a matrix called identity by descent, which basically looks at how individuals are related by the shared ancestor, not by maybe by just a bias that anyone relate to the other. And so we did drug resistance profiling because we wanted to also see what drug profiles or resistance profiles that these individuals are carrying or these parasites have from the southwestern Uganda. We also did something that was a bit interesting. We did a transmission network analysis, and this, what makes it novel is that it can identify who infected who, so you could tell the directionality of infection. And so what did we find? Basically, the low transmission sites, very low transmission sites, that is Myuko and Maziba, we found out that most of the cases in these sites are older and more male compared to those from the moderate transmission site, and also found that malaria cases in these sites were more likely to have reported travel in the last 30 days. And the main reason of travel from these areas was actually work or trade related as compared to Kamwezi, which is a moderate site where most individuals, the main reason of travel was visiting relatives. And so we were also interested in cross-border travel. And so because most of these sites are the border of Rwanda and DRC, we thought actually we would have more people reporting cross-border travel. But this was rare, though it was reported in Kamwezi, where we had around 1.5% of individuals reporting cross-border travel. So because we saw much travel being reported in the low transmission sites, wanted to know where these individuals were traveling to. And then we, if you look at the map on the right side, that is where actually the final destination of where these individuals were traveling to. So you could easily see that most individuals were traveling even far east or far north, the parts of Uganda, whereby the malaria transmission in these sites is very high. So we'd expect that probably these people also would be bringing in different strains. So we dived into the genomic data. I wanted to see whether actually genomic data is telling us or is communicating the same with the EP data. And so we found that ideally you would expect higher COI or higher diversity to be associated with the high transmission intensity. But this was different. In our case, we observed higher transmission, higher diversity or higher COI and complexity infection and effective COI in sites of low malaria transmission as compared to the moderate transmission site. And these findings would actually be, would be consistent with the travel level that we are seeing in the low transmission sites, because we are seeing much travel in these very low transmission sites. And so we wanted to see the level of relatedness and the transmission networks. So if you see those plots, they are colored by the travel history. So whether someone traveled outside the district, or whether someone traveled within the district, or whether there is no travel. So anything blue indicates that individuals traveled outside the district. So Myuka and Maziba, you would see that most cases are actually linked to travel outside the district, as compared to Kamwezi, where most individuals show no travel, because the plot is more of pink. And then you can see much of the clustering in Kamwezi as compared to Maziba, which indicates that most parasites within Kamwezi are actually highly related. And so we also wanted to know the resistance profiles in these sites. And so we are seeing most of the mutation, the K13, we did we looked at different resistance profiles, but mainly I'm presenting something on mutations associated with artemisinin partial resistance. So you could easily see that in these sites, we have those mutations that are prevalent in most of the sites. But what is more interesting is that we are seeing high prevalence of 561H and 441L in Kamwezi. And some of these mutations have been reported in Rwanda, which shows that there is actually inflow of these mutations into the country. So wanted to see this clustering that we are seeing in Kamwezi, do we have some mutations that are linked to them? So you can easily see that that large cluster within Kamwezi, it literally carries mutations that are, associated with artemisinin resistance, that is 441L. And so what do we conclude? We are seeing that epidemiological data and the genomic evidence really tell us that imported malaria drives most of the infections in the very low transmission sites, and then frequent travel to high transmission districts, mainly for work, is the risk factor. And high genetic diversity was observed in the low transmission sites compared to Kamwezi, which is more of a nodal transmission sites. Also, relatedness and transmission network confirms clustering within Kamwezi, and this reflects persistent local circulation or spread of these parasites. So detection of this C441L, a mutation which is associated with K13 artemisinin resistance, they associated with artemisinin resistance, within the largest cluster that we have observed in Kamwezi, it shows more of localized transmission. And so, what are the study implications? One, this points us to what we can say as called tailored interventions. So we need to have different interventions based on what drives transmission in these sites. For example, incorporating travel history into case investigation would really improve our detection for the imported cases, specifically for these low transmission sites. And also we need to think of targeted outreach, basically to mobile populations or mobile working populations, where we can focus on maybe return testing. If someone is moving from these highly transmission sites back to their homes, we could do more of the testing, but also we need to think of post-travel treatment guidance. And also, since we are seeing more of localized transmission within these moderate transmission sites, we can think of sustained community level control and vector surveillance to disrupt these local transmissions in these low transmission sites or the moderate transmission sites. But also we need to think of integrating genomic data into malaria surveillance across connected districts and borders. That might be also a way of tracking the imported infections, but also maintain molecular resistance monitoring, where we could track the emerging of these drug resistance markers. Yeah, so I take this opportunity to thank all the people that have contributed to this work. mainly the study participants that consented to be part of this, the entire IDRC team, our mentors and supervisors, and then collaborators, without forgetting the funders that have supported this work. Thank you.
Yes, there we go. Thank you very much, Monica Mbabazi, for the presentation. She's telling us something which is key. You know, we are thinking of moving towards malaria elimination. And the area where she did her work, southwestern Uganda, is where transmission is now low. So she's brought out key issues in those areas, a lot of genetic diversity, a lot of complexity of infection, a lot of drug resistance, and it is cross-border. What they identified in Uganda is similar to what is in Rwanda, the K13 mutations. And you see, a lot of the cases are driven by what is imported from us. So this gives us important lessons and implications for our planned move to malaria elimination. So we will need to build capacity to be able to prevent reintroduction of malaria in areas where local transmission is almost halted or when it will be halted. So, and we need such capacity to be able to track, to know which cases are imported, which cases are local. So thank you, Monica, for giving us this information. A round of clap to Monica. So, ladies and gentlemen, we have come to the end of this presentation from the panel, but it's not the end of the session. They have given us a lot of insights on different aspects of malaria. So, can you give a round of applause to all of them? Now we are moving to the Q&A sessions. Because we don't have much time, I'll require you to be very brief. You mention your name, ask the question, very brief question, and you clearly state whom you are directing the question to. So we'll take around 5 to 10 questions before the panelists can answer. Any question? Yes, somebody with the microphone. Anyone with the microphone? Bring it, please. Somebody here with the question. Right in the front here.
Thank you, Chair. Short comment and 2 short questions. The short comment is to applaud IDRC for sponsoring studies cross-disciplinary to address malaria and to see women scientists in training. I have a short question for Evelyn and for Monica. Evelyn, traditionally, and it's a non-expert question, we always look at immunology in terms of the innate immunity and adaptive. And sometimes I think we've created an artificial boundary. When you look at natural killer cells, how is the interplay between those 2 arms of the immune system? And then for Monica, the question I have for you is, I became very interested in your travel history. And in the day and age where we have mobile phones that can actually tell where you are, do you see opportunities to partner with communication companies so that as we do these information heavy sciences, we can actually tap into this rich information? You can imagine if you went to an area and you got a notification, you've entered the malaria intense zone, take precautions. So that kind of, I think that's where the science could go.
Thank you very much. Anybody with question or comment? Around the room? Yes. Next. Yeah.
Yes, thank you very much. Dr. Lubite Alan, I'm a senior medical officer from Namtumba District Local Government. I have only 2 questions. One to Dr. Kakuro. You gave a very good presentation, just like the rest of your colleagues, about when you reach to the point of using your participants. You said you explored the non-HIV positive women, but later on, out of the selected number, you used an exclusion criteria for those you had cited. But I wanted to know which exclusion criteria you used at the time. After your sample, why did you exclude the other from the final total? Yes. Then my other question is to either question or comment, but to the last presenter, that is Dambaba's Monica. When you look at the low endemic areas in southwestern Uganda, we assume that in terms of malaria, these individuals are vulnerable because in one way or the other, they don't have the long-term immunity because of the less exposure to malaria. So when we have individuals moving into high endemic regions within the country and then going back with malaria in such areas, we are still seeing a low, let me say, pan-out of malaria still, despite the malaria cases.
Can you summarize quick?
So my analysis, maybe for future research studies, or if you manage to explore, now the transporter, which is the mosquito now, in these low endemic areas, in high endemic areas, have you seen any studies that portray a difference in the ability to transmit in high endemic and then low endemic in such kind of populations? Thank you.
Thank you. Some questions down. Take that. Go ahead, please.
No. Thank you very much, the presenters. To the first presenter, you showed us that there is high prevalence of mixed falciparum, Plasmodium falciparum and malaria, and then falciparum and ovale. And yet, we tend to have RDTs that detect only one, and I think most people are initiated on that basis. What would be your recommendations for therapy in given the findings? Then for the second presenter, the deletions which you talk about HRP2, which does not confer problems with diagnosis in RDT, especially for falciparum. Now, when we use a single RDTs for falciparum, we might use it with confidence, but we have mixed, as shown by the first presenter, mixed infections. And that could be causing why someone comes, they are told they don't have malaria, but they continue to have the symptoms, so which means we need more clinical evidence. Now, what is the implication for us having these antigens, ovale, malaria, and all that to support diagnosis, given that we now know there are mixed infections, and some of these people die in private hospitals because they don't go ahead and dissect, but treat infections if there is no malaria. The third presenter, the patients, you talked about SP, the advantage of using SP, which is still good in Uganda, and probability of having adverse events in using the other arm. Now, what did you consider analysis of the patients who dropped off, especially after initiating both therapy, and what that could inform us of adverse events, or why they dropped off, or could it have caused some bias in your analysis if you didn't include it? The fourth presenter,
okay, thank you.
just one
So that will
more.
be the last one. You give the
Yes.
opportunity to others.
Maybe I go to the fifth one now. I jumped. Yes, I jumped the fourth and go to the fifth. These are very critical questions. You've told us about the clustering of resistance in the low prevalent region and then picking it from travels outside. What can we do about them not bringing that in the low prevalent region, there's also resistance in the clustering? What is the reverse effect we are going to do to make sure that we don't ensure that this resistance in low prevalence region continue to also travel with these same people going for trade into the high prevalence region? Thank you.
Thank you very much. Yes, can you have Professor Noah?
Yeah, thank you, Chair. Very interesting results. Two short questions. One to Mbavazi. What could be the role of chemoprophylaxis before travel in this country? And the next one is to Kakuru. Does this mean that malaria has stopped influencing outcomes among pregnant women? Oh.
Thank you very much. Just behind you. I think that should be the last one. No, that one, then here last.
Thank you, moderator and presenters at large. My name is Joshua from Fort Portal City, and I'm based lab. So in attempt of the nation or the Uganda to eliminate malaria, we should have better platforms for testing. Basing on that, I'm very much, and I was much triggered by the first and the second presenter. And she could have read my mind probably, because the first presenter had a proportion of mixed infection in PF and malaria, which are at times diagnosed in relation to what we are using national ways in which are supplied by NMS that are species or single species centered, that is PF, versus these deletions of histidine protein 1, rather 2 and 3, meaning that our end results, either laboratory ways we are doing misdiagnosis, which ends into mismanagement of patients. So, as a country, I know policies can be revised, they can be replaced, or even updated. What attempt has it or have we put in place to see this one addressed, such that we can have an attempt or we go towards the elimination of malaria? Thank you.
Thank you very much. Can you have here and there last? And then we respond to this, because we are running out of time.
Thank you so much, all the presenter. My name is Miriam from IRC Yumbe. I have a short question to Mbabazi. One, what factor have they contributed to the low malaria in their site? Then the second one, what lesson learned can be shared with us in some other region aiming for malaria elimination? Thank you so much.
Thank you.
I'd like to add my voice to my brother over there. IDRC, you're doing such a wonderful job in going into malaria science deeper, and I'm glad that you're presenting at both local and international fora. And also thank you for giving opportunity to young people, both young men and young women, to learn to present, because in the next couple of years, many of us in this room will be retired, and these are the trees of tomorrow. So thank you very much. My question goes to Ebo. We know that the cost of DP is, I don't know how many times more than that of SP. 1 tin of SP of 1,000 tablets is around maybe 10,000, 20,000 shillings. 1 dose of DP is 15,000 to 18,000, sometimes 20, depending on where you buy it from. What is the cost implication of scaling up your study across the country from one study site to the 142 districts? And is it attainable? Thank you very much.
Yeah, thank you very much. In the interest of time, I will say that we'll stop with these questions. So what we'll do is we'll start from the extreme end, from Dr. Abel. Please pick the questions which are referred to you, and if you have any general comment, you make it at once. So we give you, and address them just in very few minutes, in less than 2 minutes, and we'll move up to the end here. Okay, Dr. Abel.
Thank you so much. Thank you so much for those wonderful questions. One of you asked about other reasons for exclusion. So it was not only HIV. We also excluded people who were who were past the 20 weeks of gestation, people who are living outside of Busia, who are planning to move. So there are multiple exclusion criteria that we used. You can find our paper. We published it recently in PLOS Medicine, so you can go there and see the paper in details because you can't summarize it in 8 minutes. Then someone asked whether people who dropped off could these have introduced bias. Yes, lost follow-up can introduce bias, but we don't think it introduced bias in our study because the losses to follow-up were random. They happened randomly between the two treatment arms. And could this loss to follow-up be due to side effects? No, we don't think it was due to side effects. Some of them, actually majority of them, moved out of the study area. Then Professor asked whether my study is pointing to, our study is pointing to whether malaria has stopped influencing birth outcomes. No, we don't think malaria has stopped influencing birth outcomes. We think birth outcomes, there are many factors that determine birth outcomes, not only malaria, and SP could be addressing the non-malaria benefits. And then because it's also an antibiotic, people may be delivering big babies because it's an antibiotic. So we think malaria has not stopped influencing birth outcomes. It continues to influence birth outcomes. But when you use a very good drug that prevents malaria, it doesn't reduce birth outcomes compared to SP because SP has additional benefits. Then Joel asked about the cost of DP. If we are to scale it up, it's expensive, but you can't just look at just the cost. When you're calculating the cost, you look at other benefits. It's called cost effectiveness study. We need to do cost effectiveness studies. And I don't know in the audience if there are young people who are interested in conducting cost effectiveness studies. You can come to us. We have the data, and you will use, you can look at the cost involved in delivering the intervention. You look at the benefit, and then you calculate disability adjusted life years, and then you'll be able to inform us whether it's actually cost effective, but not just looking at the cost, the cost in the market. So we have all this information. If you are interested in doing cost effectiveness analysis, please, you can come and contact us. Thank you so much. But it's very important, Joel, we need to look at cost effectiveness studies to see if we can scale up DP, especially for prime gravid women.
Yeah. Thank you, Ebo. Can we move to the next? Okay, next.
All right, thank you. I think I had some few concerns about the diagnosis of malaria. I think the first question was about the mixed infections, and then when it is combined with the HRP3 deletions. So what I can just first to highlight is that when we are looking at HRP2/3 deletions, we are purely looking at Plasmodium falciparum infections, as these RDTs are very, very specific. Then when it comes to mixed infections, if you want to look at other infections, it means that we need to combine in the RDT some other antigens that are not looking at HRP2 and 3, because those ones are exclusively produced by only PF. That's why we have now RDTs, for example, which are called PF-PAN, where the HRP2, we add aldosterone or LDH so that you can see those other infections. Then there's a gentleman that also asked about what is being supplied by national medical stores, and if at all we are missing maybe some diagnosis and having improper management of malaria. I will just echo what I mentioned, is that these decisions are not yet really a threat to us when it comes to Ugandan setting, with that in our findings we found actually just less than 1% and only in HRP3 deletions, only HRP3. So HRP2 is still there, and that's actually the major antigen. Only that the HRP3 also cross-reacts with HRP2. So if we have HRP2 present, then this is not yet a worry, and our RDTs are still capable of detecting PF, and you should note that PF is the major species for us. Yes, we are seeing emerging mixed infections, but still over 90% of our infections are due to Plasmodium falciparum. Thank you.
Okay, thank you very much. Monica?
All those that have asked interesting questions. So the first was on whether I see myself or our team collaborating with the communication companies. And I think this is really an interesting concern. For example, imagine if we had like a platform, or we collaborated with say MTN or Airtel, and then if you move from southwestern Uganda where the malaria transmission is very low and probably you're heading to north, and then you just get these popping messages that please be conscious of malaria, do this, this, and this. I think that would be an interesting thing. Personally, I had not thought of it. I guess I'm still putting much focus on my PhD, but that would be something that I would consider after. Thank you for that. And the second was, I think someone asked about the ability to transmit, looking at high and the very low transmission sites. Of course, you would think that transmission intensity or the level of transmitting the parasite to an individual would be high in the low transmission sites, probably because of the low immunity, but also you need to ask yourself, why is it a low transmission site? There are other factors that are contributing to transmission, probably the ability of the mosquito to, in terms of climate and high altitude. Those also sort of like limit transmission. So I'm not sure whether there's a study that has looked at that, but we could, you can think about it. We can think about it, but I would expect still, due to the factors that are associated with the low transmission site, I would still say that transmission ability is low in the low transmission sites. And then there's a question from a professor asked about, was it about chemoprophylaxis was it before or after travel? But I would say with travel. So I think this is really an interesting thing. For example, since we are seeing that travel is driving transmission in the low transmission sites, if we have the ability to track who has traveled and where they have traveled to or where they have traveled from, then we could do either test and treat, or we just give them the medication before they travel. So I think that is a very good point to note. Sorry, I missed some questions from a lady. She was asking about factors that contribute. I didn't get that question very well. I don't know whether she can repeat that. Yeah.
We are at a sort of time, so answer what you can. Then.
I just missed her question. Maybe you can discuss after.
Okay, thank you very much. I think after here, she can get in touch with you and you can continue your discussion over lunch. Evelyn?
Thank you, Doctor, for the question. My question was on the interplay of innate and adaptive immunity. For my area of study, such as NK cells, we see they are not mainly innate cells, they are also stretched to adaptive cells because of their ability to differentiate with differences in exposure. So there's now, with more studies, we see there's a thin line between innate and adaptive immunity because of the ability to differentiate with time. Thank you.
Yeah, thank you very much. And lastly,
Thank you.
Innocent, yeah.
Thank you so much. I think I had two questions. And the first one was about the recommendations by mere fact that we are seeing a prevalence of mixed species infections in some regions of the country. And I think for those that are in a clinical setting, you are aware that in management of malaria, the artemisinin-based therapies that are used mainly target the erythrocyte stage infection. And we see that if you have these other infections, other species like Plasmodium vivax and Ovale, the main reason why they are accounting for the possible relapse is because of those parasites that are lying latent in your liver, and they're likely to be missed out. So once we are having data that can confirm that this patient, for example, has mixed species of Plasmodium, then it is important to look at a therapy that is going to cater for these other species. And that's why I think, as our colleague had already mentioned, diaretimicin piperaquine combinations will continue to be so helpful to us by mere fact that that piperaquine there can help you curb these other infections. Then there is a gentleman, Joshua, asked about the attempts that are in place to address these issues. I think my colleague Bienvenu gave part of it, where he mentioned something to do with having rapid diagnostic tests that can detect all these other species being embraced and used. But also, I also think that routine surveillance, like this study, should be something that can be embraced by Ministry of Health, that even when there are no funders from outside, it is made a routine, just like we see that in TB, in HIV, you have systems that have been set up in that if you have maybe someone that is having relapsing malaria over and over, then they can be sent to a given reference center where molecular techniques can be used to investigate more, look at the sequencing level, the different species that are there, and this person can be dealt with as a special case depending on the species that are causing the infection in them. I think that is what I can add on. I hope I've answered well.
Yeah, thank you very much to the panelists. Can you give a round of applause to the panelists? Yeah, we are rather short of time, and we would like to conclude quickly. So we had a number of presentations. Innocent talked about malaria co-infection. We realized we have mixed infections here in our setting. Luckily, the current treatment is able to treat most of the mixed infections. So know that mixed infections do exist. We talked about issues of HRP2 deletions. Luckily, the prevalence is still so low, not so much a worry, because they will they'll have impact on the current RDTs, especially HRP2-based RDTs, which we use for diagnosis. Abel talked about malaria IPTP among pregnant mothers using either DP combined with SP or DP alone or SP alone. I think the findings were clear. SP still works. It's cheap. So good to continue with it. Improves birth outcomes. DP good at mainly for the PGs, but if you combine it with SP, doesn't work so well. Picked lessons from there. Daniel talked about gametocyte production infectivity. We note that a lot of resistant parasites are coming up in northern Uganda. and the resistance is associated with increased malaria transmission. So we need better treatment. Then to the two ladies, one talked about natural killer cells and the importance in their relation with the malaria symptoms. And lastly, malaria and travel and monitoring malaria in low transmission settings. What Babazi presented, very interesting findings, important lessons to learn as we move towards malaria elimination. So thank you very much to all the panelists, and thank you very much to the audience for being a very good and engaging audience. Apologies for going slightly over the time. We started a bit late, so I hope we have not over-delayed you a bit. So can we give a round of clap to all of us? Yeah, thank you very much. So we come to the end of our sessions. Enjoy your lunch.
Okay, thank you. Can we appreciate our chair for taking us through the session? You can take a group photo with the presenters. In the meantime, colleagues, our lunch is ready. But as you go for lunch, also ensure to give feedback. You scan the code. We kindly request that we use maximally 45 minutes because we had tried to be on time for this morning, but we have now lost about 15 minutes. And we plan to start the closing session by the lecture by 2:15. So let's move for our lunch. When we break out for meals, you people spend about 10 to 15 minutes still seated, and that takes back. We can have the discussion as we are having lunch, the enough serving points, and please be mindful of your property. Let's all be here by 2:15. And the people leading the next sessions, please stay behind for briefing. Thank you.
Emma.